Good afternoon, everybody, and thank you for coming along to my talk. My name is Andrew O'Neill, and I'm the director and consultant of a company that I've set up called Nirmetrix Limited, which is concerned with near-infrared spectroscopy and the interpretation of molecular spectra, near-infrared spectra, which is called chemometrics. So in chemistry, we call sort of machine learning pattern recognition of chemical data chemometrics. Apologies for having 6 sections here, which is quite a bit. I'm just going to quickly go over some vibrational spectroscopy, some definitions and an introduction, a little bit of theory. I'll show you the portable spectrometer that I'm presenting today in the presentation. It's about the size of a desktop mouse. It's a near-infrared spectrometer, and it's actually quite good performance. So it can be used to identify things because it produces a spectrum, which is like a chemical fingerprint, and so that can be used to identify materials and products, and it could also be used potentially for quantifying the level of a particular ingredient constituent, which we might call in chemistry an analyte, within the sample matrix, so within the actual thing itself. I'll go over briefly some Perl-TK, the TK extension, which I've used to build the graphical user interface that then triggers the device to acquire near-infrared spectra, and then actually interprets the spectra and does all the machine learning in the background, so the end user doesn't have to be concerned with lots of computations, multivariate analysis, and statistics and things. I will show you the GUI that I've developed, some slides of that, and then I will show you what the GUI actually does. So I'll give you a smaller presentation on some chemometrics and machine learning, a pharmaceutical application to differentiate the NIR spectra of 2 tableted products which are on the UK market, and then just the conclusion at the end. So just to go over some vibrational spectroscopy, there are some abbreviations or acronyms in the presentation. So API is active pharmaceutical ingredient. I've not actually analysed any pure drug substance, but it's present within 2 of the products or 3 of the products that I've analysed. IR is infrared. DLP MEMS is the technology that Texas Instruments produces that allows for a miniature spectrometer to be produced. It's a device with a chip in it, which has got about 1,000 different tiny micro mirrors in it, which can move up and down, and those can reflect particular wavelengths of light, allowing the entire near-infrared spectrum to be characterized. Some of the spectra themselves require some sort of mathematical manipulation to correct them, because there can be non-chemical sources of variation in the NIR spectra, things like curved backgrounds and spectra having variable baseline offsets, so jumping up and down on the y-axis. So we may do things like extended multiplicative scatter correction, EMSC. GUI is obviously graphical user interface. InGas is the type of semiconductor. It's a mixture of indium and gallium arsenides, and that is the thing that is sensitive to NIR light that then produces, electrons or things like that can be detected. Multiplicative scatter correction is another type of scatter correction. NIR is near infrared. PCA stands for principal components analysis, and that's one of the main ways of doing dimension reduction. So you can reduce the dimensionality down of a spectrum which might have a few hundred different wavelengths in, down to a few abstract vectors, and then a spectrum can be represented as a data point in a small pattern space. PLSR is similar to principal component analysis. It's another multivariate method, but it's a regression method that allows you to build a quantitative model to predict the amount of a particular ingredient or analyte in the sample. Perl-TK, that's obviously used for the TK extension for the GUI. PTFE is a white reflectance standard that is used in NIR, and R is the reflectance, which is the actual thing that's measured. That is the light that is reflected back from a sample such as a tablet, and it's measured relative to the light that's reflected back from PTFE, which is the non-absorbing standard. So it has values between 0 and 1. SG stands for Savitsky-Golay. It's a type of smoothing filter that can be applied to the spectra and also derivatized to resolve overlapping absorption peaks. And SNV is another type of data preprocessing, similar to MSC, etc. T-squared here, which is just missing from the slide. My apologies there because I've used LaTeX. It's chopped off T-squared. T-squared is the distance in the pattern space that we use to determine whether a particular projected spectrum falls within the usual cluster of data points. And if this value exceeds a critical value, then we can say it doesn't. The spectrum doesn't belong to the class and reject it and say it's not a match. So many of, if I move on to the introduction, many of today's methods of instrumental analysis produce vast amounts of data, lots of measurements of samples, and at lots of different, say, wavelengths or different variables. And there could be hundreds or thousands of variables in each piece of data that's captured, and then there can be thousands or millions of spectra that might be measured. And it's just not possible, really, anymore to, for a person to properly interpret lots of spectra. Near-infrared spectroscopy is a type of spectroscopy that generates this type of data. It was discovered in 1800, but largely ignored because the peaks tended to be sort of broad and overlapped, and people thought that it was more or less a sort of a rubbish region of the electromagnetic spectrum, so it was ignored. But the actual vibrations themselves arise due to higher energy photons that match the vibrational ground state to the first, second, and third overtone vibrations. So it's sort of higher energy vibrations than the traditional mid-infrared spectrum, which would be transitions from ground state to the first vibrational level. It tends to have what's called a lower molar absorptivity than infrared spectroscopy, and this is of an advantage because it means that the light can penetrate into a sample like a tablet and not be completely absorbed, and therefore, because this is a type of reflectance spectroscopy, we need to get light reflecting back onto the detector. And if we were using infrared spectroscopy, all the light would be absorbed, nothing would be reflected back, and we would max out the detector. We would basically have a flat line with no sort of chemical signature in it. But we don't get that with near infrared. We get light penetrating into samples and then reflecting back. That's called diffuse reflectance, and we can make use of that now to identify things. The other advantage, of course, with that type of with this particular sort of physics is that we don't have to grind samples up and dilute them down like you might have to do in infrared spectroscopy. We can analyze things intact, like entire tablets, and measure and characterize them within seconds. So typically the spectra have got broad overlapping absorption bands, and as I say, the baselines may not be particularly flat. They may have linear and curvilinear trends in them, and these relate to the scattering of light, not to chemical differences in samples. So we need to correct the spectra sometimes and to remove these linear and curvilinear trends from the spectrum. So the vibrational, this slide shows potential energy, so the energy in a bond, and if we have R equals 0 here, so the bond isn't stretching or compressing, you can see the change in the vibrational quantum number as the bond absorbs energy, goes from the ground state to the first vibrational quantum level for infrared spectroscopy. But near infrared is these overtones, so they're higher energy transitions. They're multiples, effectively, of this vibrational level. And as you can see, they're discrete levels. There's not a continuous distribution of possible absorptions. It's quantized. Bonds themselves can either stretch or bend. So this is what we're looking at with the spectroscopy. So you can have symmetric or anti-symmetric stretching, and you can have various different types of bending of bonds as well. As I briefly touched on in the definitions, there are different types of scatter correction to correct the baselines. the linear and curve trends within the baselines of the spectrum. So one is detrending, which is literally fitting a least squares quadratic into the spectrum, subtracting that away. Standard normal variate, multiplicative scatter correction, and extended multiplicative scatter correction are other things that can be applied, or a sort of smoothing and differentiation filter. And popular ones in signal processing are Savitzky-Golay filters. They can be used in near-infrared spectroscopy. So moving on to the aim. The aim of the work that I did here was to use Perl for two purposes. One was to control another language that I use called Octave, which is like MATLAB, and to get Perl to generate lots of different mathematical models for me, to save me having to do this manually. And then I used Perl to develop a graphical user interface to control a handheld spectrometer, which is about the size of a desktop mouse. It's a near-infrared spectrometer, and I acquired one of these things, and it came with software that runs on a smartphone. It generates a spectrum, but didn't actually do any interpretation with it. So I thought, why not, sort of write a GUI and then do all the chemometrics and stuff like that, and turn it into something useful. So moving on to the spectrometer, this is what it looks like. As I say, it's about the size of a mouse. You can see here there are two tungsten halogen lights in it, a bit like in a torch, and they shine light. And you would put your sample onto this quartz sapphire window, which is transparent to near-infrared radiation. And about 8 seconds later, the device will have measured about, say, 32 spectra, and taken the average of that as representative of the spectrum of whatever the product is, and then send the data to the PC. Here I'm using it in a Bluetooth, a wireless mode, but my GUI requires this to be a wired USB connection. So that device itself is made by a Taiwanese manufacturer called InnoSpectra, and it's based on a Texas Instruments spectrometer design, and it's model NIRSG1. It's got an indium gallium arsenide detector in it. It's an uncooled detector, so it only the wavelength range is 900 to about 1700 nanometers. Different modes of operation of the micromirror. Linear, which is called column. There's a Hadamard transform that can be applied, which is similar to a Fourier transform. And a slew scan where you can just literally move to a particular wavelength. So I've used it in the linear mode, which I think Texas Instruments recommend for this particular product. And I had it connected to my PC, just an Intel next unit of computing, a NUC 8th generation i7 with 64 gigabytes of RAM, about 2.8 gigahertz. I mean, it's overkill for this device. You don't need something as high spec as that. So if I move on now to the Perl TK extension and the GUI, got to have reasons for building a graphical user interface. Well, it's a convenient way to get lots of information into Perl scripts. Saves having to type lots of different things on the command line. And it can add flair and professionalism to an application and make it look quite slick. And also it's useful where you've got to have many decisions and lots of input from the user are required. So it just makes life easier, I think. So Perl TK is the TK extension for Perl 5 for creating a graphical user interface. And TK itself includes several different modules. It's object-oriented, and you can create windows with it, which are, I think, called widgets, buttons, lists, and text and other things like that you can put into it. And it was ported to Perl by Nick Simmons from TCL at TK. So if you wish to test whether you've got the TK module installed on the command line, you just type Perl -E, use TK. And if you don't get anything when you hit return, then you've got it installed on your system. So to create a widget, then it's the usual sharp bang, use bin Perl, use TK, and then you have to create the main window widget, which is the first window, and it's the top level widget. So $MW equals main window, and then type the rest there, new, etc. The next widget that you'd have to create is a frame widget. So $frame equals main window and then frame. And that's the container to hold other widgets. It's usually invisible. There are two separate commands though for each widget. One creates the widget, but you also have another command to display a widget. So you use pack, tend to use pack, but I think there are other commands as well. But $widget pack, and then should be displayed. Well, you've got to have what's called an event loop. So GUI input is defined by events. So it's a combination of user input from keyboard and the mouse, like pushing the left mouse button or moving this cursor, this pointer around the screen. So in Perl-TK, the event loop is initiated by calling the routine main loop. So here is an example, a very simple example, which I've taken from the book by Walsh, Learning Perl-TK, released back in 1999. So this very simple set of this code here, 1, 2, 3, 4, 5, 6 lines of code, and you've got a very simple widget here with the done button, which when you press it, the GUI sort of disappears. Okay. So moving on to my GUI, I've set it up to have to make it a very simple looking GUI with 3 large buttons, something that could potentially be used on a touch screen, so that a user doesn't have to use a mouse in order to control the spectrometer. So there's 3 buttons: one to acquire a scan of a sample, but also the background reference scan of that PTFE standard that I mentioned. That has to be acquired. That's quite important to acquire that first. You need the background scan at least once a day. That has to be done. And then there are 2 other buttons, one for qualitative analysis. That is if you want to simply identify something from the spectrum that's generated of the sample. But there's also a quantitative analysis button if there's a model built to quantify a particular ingredient or constituent within a sample that's analyzed. And the GUI does a bit of housekeeping. It makes sure that the input is correct from the user, and that the model files that are on the machine are actually sort of bona fide models. They've got everything in them that is required. So this is the GUI that I've developed. So on the left here, we've got the window with a scan button, which brings up this scan window. And as you can see, a reference scan on this particular day wasn't acquired. So the user can only acquire a reference scan of the white reflectance, background reflectance standard. Acquiring a scan of, say, a tablet or some other product, it isn't possible. It's all grayed out. Once the background scan, which is this broad thing here, has been acquired, I've moved on here and measured the spectrum of the tablet. This is something you can buy over the counter in Boots. I think it's a pseudoephedrine tablet, so it's a nasal decongestant tablet. This is the spectrum of that tablet. And here is the qualify button. Once that's pressed, this window appears. The pattern recognition, the multivariate analysis of this particular spectrum has been done, including baseline correction. And the spectrum has passed. So it's passed on 2 types of pattern recognition statistic. Hotelling's t-squared. It's below the critical level, the 95% critical value, and also the residual, what's called the Q statistic, which is the residual standard deviation of the spectrum, the bit that's not actually used. That has to be below a certain statistical threshold, and it is. So overall, there's a pass. Now, with this same spectrum, it's possible to qualify it against another model, and if I was to do that, it should fail. And on this slide, you can see here the very same spectrum, and I've qualified it against a chloroquine, which is a antimalarial model, and it's failed, which is correct, which is obviously correct what it should do. A Boots pseudoephedrine tablet shouldn't match a chloroquine antimalarial tablet spectrum, so it's correctly rejected it. And if the user tries to qualify something and hasn't highlighted, they get that window, no model selected. And if somebody puts a rubbish model in, or just makes something up and adds it into the directory, which has got all the models, the program will check the contents of the file, and if it doesn't contain everything it needs to, returns corrupt model file. So that is what the GUI does for the end user. So the end user doesn't actually do any statistics or any machine learning, but what is done under the hood of the in the background of the GUI. What is the GUI actually doing? Well, it's doing the pattern recognition for the end user. So here is an example where I can briefly show you some qualitative method to identify spectra of Silas tablets, and this model is validated using spectra of Viagra tablets. So they're different products. They're made of different fillers, different excipients, and the active ingredient, which is within each tablet, is different. Silas contains tadalafil. Here's the 3D structure of tadalafil. These little white balls here represent hydrogen atoms, and the grey ones, I believe, are carbon. The blue one is nitrogen. Red is oxygen. So anything with a white atom attached to it is likely to be NIR active. So NIR, as I should have said earlier, it looks for it measures absorptions of heavy atoms connected to hydrogen atoms. So it's going to be carbon, hydrogen, nitrogen, hydrogen. There may be an oxygen with a hydrogen attached. But you can see here we've got we should have CH groups coming up, and then maybe an NH group, etc. So the pattern recognition, I did some baseline correction, scatter correction, standard normal variant SNV, and then I reduced the dimensionality down of the spectra from 228 wavelengths just to 2 principal components. So quite a substantial reduction in dimensionality of the data. And then, so each spectrum becomes a point in a 2-dimensional space, so it can be plotted in Cartesian coordinates. So they're called scores vectors, and what we're measuring is the distance from the center of the cluster. That's called the t-square, the Hotelling's t-square distance. We can put a critical value on that and draw an ellipse, a probability ellipse around. Anything that falls inside the ellipse would be identified as being a class member, and anything with a distance that's greater than the critical value would fall outside the ellipse and would be rejected as a non-class member. So here's what the spectra in red of Silas look like, and the spectra in blue of Viagra tablets. The first thing to note is there's 2 classes within both Silas and Viagra, and that's simply because the scattering properties are different. Each tablet has different markings on one side to the other side, and that causes this baseline offset along the y-axis here and here. When we apply the standard normal variant transform, which subtracts the mean away from each spectrum, it draws these spectra closer together, creating, if you like, a single group really for each. But it also corrects some of the kind of curvature in the spectra. So as I said, the qualification of a spectrum then it involves projecting it down into the principal component pattern space and measuring the distance from the center of its cluster called t-squared. This equation here gives you the critical value for t-squared, so it's based on the F distribution. K is the number of principal components, N is the sample size for the model, F is the critical value from the F distribution for K, and n minus k degrees of freedom with type 1 error alpha of 5%. And this is essentially what you get. Obviously, it's not necessary to plot this when you're using the GUI because the GUI can do it all based on the numbers that come out. But for people, it's good to have a graph just to show what's actually going on. So here we've got our probability ellipse, 95% probability ellipse, which encircles the projected spectral scores of Silas, and they all fall inside that ellipse. And the Viagra spectra are projected into this pattern space and fall outside the ellipse. So if I was to run this model on the GUI and measure one of these Viagra spectra against a Silas model, it would come back with that red GUI window and say it doesn't match. So that's essentially what the GUI does. It takes about 8 seconds to do a measurement, so it's very quick. And the GUI it's virtually instantaneous in the actual processing. So the GUI, basically, it's been quite useful for giving a seamless user experience for qualifying the products. And Octave has been quite useful for, well, the use of Perl and Octave has been useful for churning through lots of different models and finding out the best model to use for identification. I was able to correctly identify the spectra for the examples shown, and also it'd be possible to repeat this process with the other product, and I would imagine the results would be the same. I'd like to end just by acknowledging some people. So Lee, I'm very grateful for your help. I don't know if he's here today, but I saw him earlier. So very grateful to Lee Johnson for accepting my talk and for being very helpful in emails with me. Also like to thank Will Braswell, who's the founder of Auto Parallel Technologies Incorporated, Brett Estrade, who's a contract Perl programmer at Acutus Computing, data consultancy, and Mark Perry, who's reviewed some abstracts and things for the Science Perl Journal, which is something that we have recently established as a group of independent Perl programmers, and happy to take any of your questions.